Thank you very much for your kind introduction. Of course, it's a great pleasure and honor to speak at this exciting meeting on this place. Now, ladies and gentlemen, as you all know, today almost all life on Earth appears to be adapted to moderate temperatures, that's between 10 and 40 degrees centigrade. This is also true for the smallest living beings, the bacteria. A few heat-loving bacteria have been known already since a very long time. In 1862, it had been Ferdinand Kohn who had reported of green layers of cyanobacteria which grew at 55 degrees within the hot springs of Karlsbad, Czech Republic. In 1972, this is 110 years later, James Briley and Tom Brock discovered Sulfolobus, a microbe thriving at 75 degrees within acidic hot springs at Yellowstone National Park within the USA. Louis Pasteur had observed that bacteria of the human environment are safely killed by heat treatment at 80 degrees and above. The pasteurization technology is based on his findings. On Earth, the first chemical traces of life by isotope fractionation, which are pretty safe, had been discovered within 3.8 billion year old remains of cratons, for example, El Pilbara, Australia, and Isua, Greenland, by Brazier and Szydlowski. That's what the early Earth may have looked like at that time, 3.8 billion years ago. Of course, nobody could take a photograph, so this is also an animation only. Of the early Earth, that's may have been the paradise of the microbes, a hot, dirty, humid world, generally anaerobic, a lot of volcanism because the crust was very brittle, and with still, this had been the end of the great bombardment, and so there have been still serious impacts which may have contributed to the distribution of microbes existing at times, at least among our solar system. I come to this point a little later. Only superheat lovers similar to the hyperthermophiles discovered by Wolfram Zillig and myself, which exist on today's Earth, should have been able to thrive and survive in such an early Earth scenario, and they may have become even our ancestors. So hyperthermophiles, as a rule, grow optimally at 80 degrees and above, and usually they don't grow at 60 degrees or below. I come to this point again a little later. At the University of Regensburg, I have a strain collection of about 1,500 different strains of hyperthermophiles, among those even isolates surviving autoclaving at 121 degrees centigrade for hours. In my talk, I want to focus on the most extreme of all, the hyperthermophilic archaea, which grow up to the stability limits of the building blocks of life, like amino acids, nucleotides, lipids, and so on, as you have heard already. As you will see later within the tree of life, these microbes cover all the deepest branches, indicating that hyperthermophily had been a very ancient feature of life. My hunting for novel archaea began in 1979, shortly after the discovery of the archaea by Carl Woese. At that time, it had been the common view that extreme thermophiles, the few which had existed at that time, they called it caldoactive types, derived from mesophiles, so they were derivatives. The mesophiles should have been first. Tom Brock's sulfolobus had been known already at that time, which thrives at 75 degrees, was taken as a very rare curiosity among the pseudomonads. So it was completely misclassified, but in reality sulfolobus, as you all know, is an archaeon. Its sulfolobus grows aerobically. It's an aerobic sulfur oxidizer. And this was taken as proof that an aerobic lifestyle is essential because it yields, at least in E. coli, more energy to survive, and much lower growth temperatures at that time had been found within the strictly anaerobic methanogens, and this was seen as a further proof that never high temperature organisms could grow at anaerobic environments. High salt concentration, not very high salt, but seawater, was taken as an additional stress hostile to a hot lifestyle. Well, I never cared about prejudices. So on our first trip to Iceland in 1980, Wolfram Zillig and I, we wanted to find sulfolobus, possibly existing sulfolobus viruses, in order to transcription studies this organism, because it has an archaeon. And we found some very exciting environments. This had been in the Krafla area. When we went there, it was 3 days before a fissure eruption occurred there. Whole lumps of mud had been catapulted into the air, and steam, hydrogen, CO2, and H2S escaped there. And I had a field microscope with me, and in our field microscope, we discovered some organisms there. Well, this environment were really, shortly before this eruption, boiling mountains, as you see here, with all these mud holes. You had to be really careful not to fall in. I poured in resatsurin, a redox dye, because I wanted to see, is it aerobic, is it anaerobic? The redox dye resatsurin was immediately reduced from blue to colorless. That means these environments were strictly anaerobic. And in the field microscope, I saw things never had been seen before. Huge, in microscopic scale, of course, antler-shaped branched cells, 100 micrometers long, and with true branchings. And they were up to 10 to the 7 per milliliter. So these were natural bioreactors, practically, which had never been cultured before. Now, later in the laboratory, Wolfram and I did growth experiments, and we found out that these were strict anaerobes growing up to 97 degrees, far higher than the aerobic sulfolobus. And later, we described those groups of organisms, there are several, as the thermoproteales. Well, I had the idea also, could there be organisms even living up in superheated water above 100 degrees centigrade? Of course, not in steam. So the idea was to look at the bottom of a water column, it's a sea, for where it's hot enough for such possibly existing organisms. So my colleagues and everybody said, you're crazy, you will boil in your own soup if you dive there. And because I wanted to go by scuba diving, and otherwise they said it would have been found a long time before. It's so ridiculous. Some of these colleagues later began to work with hyperthermophiles also. Well, could this I went, therefore, together with my young family, spent our family holidays here. It was nice holidays. I always went down, took samples, and there had been whirlpools at the surface, and it was not deep at all, up to about 10, 15 meters. So it was easily accessible by scuba diving. You had to be just careful, and you had to be damn fast. So here is one of these waters coming out with 105 degrees. And I realized already at home in the bathtub, I tried it, how close I can come to the very hot water underwater, and just regular rubber gloves protected you for several seconds, which was enough to take samples with this syringe with an enlarged inlet. And here I took samples from up to 105 degrees. 10 meters raises, the pressure of 10 meters raises the boiling point of water up to 121 degrees, which would have been more than enough to find such an organism on this site. In the lab, from these samples at 105 degrees under strictly anaerobic conditions, we did a lot of experiments there, but after 3 months, a novel marine archaeon, the Pyrodictium occultum, grew up, and this is the original culture bottle. It was pressurized with hydrogen and carbon dioxide, strictly anaerobic, and here you see the sulfur, because there were a lot of sulfur crusts where I took the sample, so I added sulfur, and this is artificial, or it's seawater. And what you can see here are these cobweb-like structures which overnight at 105 degrees and then at 110 even, it was the upper border, began to grow here. And when we looked in a normal microscope, you could not see why this is these are networks, but they're much too thin. But in a high-intensity dark field microscope, you saw that these cells, which are over-illuminated, they are disc-shaped, they are connected by these little tiny hollow tubules. Here is a more recent electron micrograph, and you see these disc-shaped cells of Pyrodictium, and these enormous tubules which extend. So it's almost like a multicellular prokaryote. The tubules are hollow and penetrate into the cells, come into contact with the membranes, go out again, and these whole networks are centimeters, and they are also, we found them later when I had been invited to use Alvin. They also inside of black smokers, and everywhere are thermal gradients. So I have some speculations which about the function of this I could talk about in the discussion later. For the first time, the ability to grow in superheated water under slight overpressure up to 110 degrees was therefore observed at this organism. With this experience in my mind, during the following years, I visited high temperature areas all over the world and isolated and tried to isolate high temperature organisms from there. For example, as I had mentioned already, here I take samples from a black smoker at 21 degrees at the East Pacific rise. Holger Janos from Woods Hole had invited me to come. This was off the coast of Mexico in a depth of 2.5 kilometers. The hot water escapes from the Earth's mantle, as we have already heard. And the deep sea is very cold there, about 2.8 degrees. And so this is like a huge, as a model, maybe a huge macaroni. And there's a central heating with about 400 degrees by the escaping fluid. This is sterile if you take it in the proper way. Holger Janos had a nice equipment there, titanium syringe, call it smoker poker, which we put into this and sucked in it's not smoke, actually it's precipitating minerals. and there's a cooling from outside, so within these walls there are enormous temperature gradients, and that's exactly where we found some exciting hyperthermophiles there. Well, but also, I also looked for seemingly unsuitable places to find hyperthermophiles, like here at the North Slope in North Alaska. It's a Beaufort Sea. That's where some of the American oil drilling is going on, and I had been invited by BP to go there. These are the wellheads, and there is a 400-meter permafrost, and there is no volcanism. It reminded me on maybe on Mars today. The crude oil is situated in a depth of about 4 kilometers and has an in situ temperature of 80 to 100 degrees, and there was always some reservoir souring going on, and nobody really knew what was going on. It was always attached to chemical reactions. So I had speculated from some organisms I had already isolated from the sea that there may be microbes on work, hyperthermophiles, and therefore they had invited me to come and take samples, the same here as at the North Sea at the Thistle Alpha oil platform. So here I tapped crude oil, so still at the surface, it takes only half an hour to come up. It was oil, water, gas mixtures, and it was still hot there. Well, back to the lab. Yeah, even unexpected for me, we got an overwhelming variety of hyperthermophiles. Some of these hyperthermophiles I knew already from the sea, like Pyrococcus, I just mentioned the names, Pyrococcus, Archaeoglobus, and Thermococcus, so they came up with concentrations in the upcoming fluid of up to 10,000 cells, you see it here, per milliliter. And these were only the organisms which became released. You could call them pioneers or whatever. They wanted to go to another environment, or they were washed up. So there must be enormous masses of microbes down there. So I think this is, and that's also what I wrote in this paper, when I published it, it's not the evidence for a coherent biosphere, but it's evidence at least for pockets of microbes which are growing deep down. This is 4 kilometers. This was more detailed data from the Thistle Alpha platform. And the organisms which I got there were unable to grow at 60 degrees or below, so they could never grow at the surface. They had to grow they must grow, therefore, in the depths. And the optimum, they grow fastest between 80 and 102 degrees. Well, the samples were always brought to the laboratory in order to do growth experiments and characterize the organisms there. Well, during the last years, there have been many of them. I cannot go into any details, but I just want to mention a few of them, of the newly discovered microbes, like Thermotoga, Thermocrinis. These were brocs, live in boiling water. They grow in the lab up to 89 degrees, which is the boiling point of water in Yellowstone Park. Aquifex, bacterium growing up to 95 degrees, highest growth temperature for bacteria. Acidicanus, Thermoproteales, I had mentioned them already. The Staphylococcus, the Pyrococcus, which is used very much in molecular biology now, and the Archaeoglobus, a sulfate-reducing archaeon, one of the major H2S sources down in the oil wells, for example, in the oil platforms, because they pump down seawater, and the microbes which contains 30 millimolar, as you know, sulfate. So this was ideal for the microbes there, because they found their electron acceptor, they pumped it down, and therefore this horrible H2S accidents where people came to death even. Now, methanopyrus candleri is a methanogen growing up to 110 degrees, and therefore this was evidence that even at 110 degrees there's biological methane production. Well, this grows optimally, that means fastest at 100 degrees, this methanopyrus candleri. Now from the mid-Atlantic ridge, a depth of about 4 kilometers, I isolated from a beehive smoker. It's here, and this is the claw of Alvin. I isolated Pyrolobus fumarii with the highest growth temperature in our hands so far. No growth at 90 degrees because it's still too cold for these organisms. That means the organism is adapted to higher temperatures, and it grows up to 113 degrees with an optimum around 106 and survives at least 1 hour or 2 hours autoclaving at 121 degrees centigrade, which kills otherwise even spores and all life. For mass culturing, here I think this is a temperature dependence of this organism. The doubling time, the faster they grow, the shorter the doubling time versus the temperature. This is for this Pyrolobus fumarii, and you see it begins to grow at 95 degrees with a doubling time of 6.5 hours, and at between 100 and 105, it's adapted, it has only 50 minutes doubling time, and this organism is a nitrate ammonifier. It's reducing nitrate with hydrogen gas forming ammonia, and at 110 degrees, it grows still with 8 hours doubling time. So really a true hyperthermophile, not only surviving, but growing there very fast. For mass culturing, based on chemical stir tank reactors, together with a Swiss engineering company, I developed with them novel high-temperature fermenters. The Archaea Center, now operated by my successor, Michael Tom, because I had to stop based on age, enforced retirement. So he operates this. There are 8 fermenters, glass line, steel and titanium fermenters with a total volume of 1,100 liters, where we can simulate the harsh growth conditions of such organisms, superheated seawater, sulfur, and so on, H2S. And the cell masses go to scientists all over the world, still nowadays from there. In their physiological properties, the growth requirements like temperatures, pH, ionic strength, and redox potential, and energy sources, hyperthermophiles are specifically adapted to their hot environment, very similar to the what already Lynn had shown in her talk. Now comes a horrible table which should say this, but please don't read it. The only thing I want to show you is about the temperature. There is a minimum growth temperature, the optimum and the maximum, and similar to E. coli and bacilli and regular normal bacteria, the difference between the minimum and the maximum is always 20 to 25 degrees centigrade, and the optimum is shortly before the maximum. So, and there are hyperthermophiles like Sulfolobus, Sulfolobus, I had mentioned already. There are more Sulfolobus now, and also some isolated by my group and others. and they grow in the acidic range from pH 1 to 5, and the others are practically more or less mesophiles, or mediocre neutrophiles, excuse me, and there are practically no hyperthermophiles which grew, to my knowledge, which have been isolated from the alkaline environments. And they are mainly anaerobic. Some of them can also use oxygen, but in a micro-aerobic lifestyle with very, very low oxygen and high oxygen is still killing these organisms. Many hyperthermophiles are strict or facultative chemolitho-autotrophs. This word has already been mentioned by the talk before me, and therefore, Bob, I want to mention it. That means CO2 is the only carbon source of these organisms if they are strict chemolitho-autotrophs and they use chemical energy sources. And here are some of the energy sources what they use. Hydrogen is the most powerful and most widely used energy source of all these organisms. And it's also, as you know, present in the volcanic environments, but also in the rocks and so on. And then it's always redox reaction, so the hydrogen has to be oxidized, but this would be only if there's oxygen, and then it's an aerobic or microaerophilic organism, and then water is the end product. But mainly they use in the anaerobic environments other chemicals. There have been a question already yesterday what kind of things they are. There could be carbon dioxide. The carbon dioxide, the end product is methane, or oxidized iron magnetite is formed, or from sulfur or sulfate, hydrogen sulfide. These are the big stinkers, of course, and in the laboratory when I grow them in these big fermenters, I always smelled in the morning if the organisms are growing. But one has to be very careful because it's a very, as you know, very toxic compound, at least in high concentration. The nitrate can be all the way through either to nitrogen or all the way through to ammonia. And otherwise, for example, aerobically pyrite and sulfur can be oxidized and to sulfuric acid and iron sulfate. This is called ore leaching. It has also a technical application. Otherwise, the requirements are very simple: water, heat, trace minerals, and that's all. Of course, liquid water. All cell components, for example, the proteins, have to be heat stable. The organisms are too small to have any insulation. And this is just an example of an alpha amylase of Pyrococcus vosii. This Pyrococcus grows fastest at 100 degrees. This gene was cloned in E. coli and then expressed, and also this, so it's purified, and it's always easy to purify such enzymes in E. coli, just boil it. And this is the most important step. And you see the activity during 6 hours, it's stable at 100 degrees. And even this protein, this enzyme, at 120 degrees, there's still some activity after 6 hours, and at 130, even at half an hour, there's still activity. So these are very, very stable enzymes, but this is not only for the enzymes, it must be for the nucleotides and for the membranes, of course, also components. In the small subunit rRNA-based tree of life, without exception, I've mentioned this already, the hyperthermophiles this is due to Karl Bose. Unfortunately, end of last year, Karl has passed away, and this was when he had obtained the Crawford Prize in this building. I think it had been about 8 years ago. And this tree is based on the protein synthesis machinery, on the, these are the ribosomes. There are a small subunit, a large subunit, and a small subunit, as Karl Woese had found out, is a ribosomal RNA with about 1,500 bases. And these bases, 80% of these bases are identical in every living organism because they are essential. And 20% are variable, as Karl Woese found out, and that's on what this tree is based on. And there are three, by the way, this is not the origin of life, because all these organisms we have in this tree had already the protein synthesis machineries. They had already at least ribosomal RNA. So the origin of life would be at least 4 floors below that. And what you clearly can see, there are the bacteria, including, of course, also the, all kind of bacilli and so on, pathogens, and there are the eukaryotes, as known, which have the nucleus branched by a membrane, and what Karl found out, there are organisms which look like bacteria, which are no bacteria. These are the archaea. In their molecular properties, they are more like eukaryotes than bacteria. These archaea have 2 major branches, Karl said kingdoms, the Crenarchaeota or sulfur archaea and the Uriarchaeota like the methanogens. These are the 2 major, and there are deeper ones which we cannot clearly say belongs it to this one or that one. I come to this point a little later. Hyperthermophiles are the bulky lineages, bulky red lineages, and as you can see, among the bacteria, there are few, but there are some hyperthermophiles. I'm very happy that we have found this in my lab. And within the archaea, there are many, and they're always the deepest branches without any exception. And so, yeah. And one point I wanted to mention also, there are no hyperthermophilic eukaryotes. As you can see here, the eukaryotes come out from, this is the branching point of bacteria and archaea. The eukaryotes come out of the archaea, but no hyperthermophiles. It's completely unknown why, but this is, I have personally also looked very carefully for eukaryotes in environments where the hyperthermophiles are there. I found eukaryotes, but they were not growing at very high temperatures. Some were feeding even on hyperthermophiles, on methanogens and other things. Finally, as had been mentioned shortly in the introduction, let me show you some last findings of my lab. The first organism, which had been mentioned, which I named Nanoarchaeum equitans, have been isolated from the Kolbensee hot submarine vents north of Iceland. It requires a host. This is, say, a very, very tiny organisms, only 0.4 micrometers, the size of viruses like chlorella viruses, big viruses, and the Nanoarchaeum grows at the surface of this big host, the Ignicoccus, the fireberry, which grows on hydrogen and sulfur, so again a big stinker. And so this Nanoarchaeum has the smallest genome known, 490,885 base pairs, and when we looked onto the genomes of both organisms, the host and this tiny guy, have been sequenced And when we looked to the genome of this tiny guy, we were very surprised. These are the brownish thing is information storage and processing. But look at this green one. There are practically no metabolic genes. This organism has no metabolic genes, while these are other archaea, hyperthermophiles, which all have, of course, their metabolic genes. So therefore, as a consequence, this organism is unable to grow by itself. So it could be simply a parasite, but I will tell you that this is not the case. This is a 3D reconstruction of this association of the two organisms. The Ignicoccus host this is Ignicoccus host, and this golden thing, this is a Nanoarchaeum equitans, which is sitting there attached. And the Ignicoccus hospitalis, as my former co-workers have found out now, is unique for all living organisms. It has two membranes, almost like a eukaryote, but the outer membrane harbors the energy conservation, for example, also the ATP synthesis machinery, but there is an inner membrane which comprises only the ribosomes and the DNA. And so these little guys obviously sit there and are stealing the ATP of its host. And in addition, there's a lot of vesicle transport going on, which is also unique for prokaryotes. And obviously by that, they get their cell components, these little guys. At present, we are still far away of a deeper understanding of the system. You could say it's a sick organism with all these parasites, but just the opposite seems to be the case. These guys, they live, as we know now, by work of other people, which looked on the black smokers. They grow on the rim of the black smokers, on the hottest places thinkable, and it's hard to be thought that this is a sick organism. And here is a gross alive dead stain of a culture which we grew in our big fermenter. And you see here the central, it's almost like a sunflower with its leaves, the leaves out there. This is a tiny Nanoarchaeum, and in the center is the Ignicoccus. Red is dead and green is alive. And you see, they live with their host and they die with their host. And this makes no sense for a parasite. A parasite always says the rats are leaving the sinking ship, of course, and they want to find another host. This is not the case, and therefore, with this organism, therefore, we don't understand this so far. And the Nanoarchaeota are no curiosity, but their 16S ribosomal RNA was so different from everything that it had been completely overlooked. All environmental probes did not work with that. And now, of course, after their cultivation, it could be done. And here you see some of the Nanoarchaeota diversity from all places like Kamchatka, Indonesia, Iceland, China, Yellowstone, and so on. And there are more, possibly even in cold environments of this system. Now, finally, just how is it?
You're doing a great job, and we love every minute. But if you don't mind, try to wrap up, and we'll have time for a couple questions.
Of course, yeah. Maybe I leave the last one. I just wanted to say, since 15 years, by Norman Pace and Jody Sayhead, found by environmental studies there is only the 16S ribosomal RNA. The organism was completely unknown. There is the deepest branch among the archaea, and nothing was known instead. And we were able recently to get the organism out. Here it is. We call it Coarchaeum cryptophyllum. And this is an electron microscopic image. This organism has 0.16 micrometers diameter. It's a huge, long, needle-like organisms, however, with blunt ends. They are completely invisible in the regular light microscope. And therefore, it was really the toughest nut in my life to hunt for these organisms. And they are very exciting because in their genome, they have genes from the Euryarchaeota and from the Crenarchaeota, from the two kingdoms of Archaea. So they must have branched off before that. They must be very, very early organisms in the history of life. Well, certainly our findings are only the tip of a very hot iceberg, and organisms with similar properties may even exist on other planets and moon whenever volcanism and liquid water come together. At least within our solar systems, they could have spread a long time ago, powered by impacts, for example, at the end of the Great Bombardment. Thank you very much.